progesterone, and then we start having those, because of the types of negative feedback between progesterone and estradiol, either with FSH or with LH, it would create, something that, if they are, especially progesterone, present in circulation, you're almost never going to have ovulation, because it's going to drop LH concentrations or LH pulsatility to a point where the follicle can no longer develop. Then, very, very last thing that we went through was the presence of prostaglandin. That at some point, prostaglandin will be produced or synthesized by the uterus, and their most important role in all of this is to cause luteolysis, which is basically killing the corpus luteum. Basically means killing progesterone, and with that, it would go on to be able, the follicle would be able to then continue to develop until a point where estradiol would be much, it would go in much greater concentration. And at some point, that is actually another point where we said, yes, you've been telling us that estradiol is super bad, for the feedback, but then I specifically mentioned that it was important to bring back those different localizations in the hypothalamus, different centers of GnRH release, which one of them, which we call pulsatile center, has a very strong negative sensitivity to estradiol. So as estradiol binds there, FSH or LH goes lower, especially FSH, but it goes lower. But as estradiol goes up to a certain threshold or beyond this threshold, it impacts a different center, and this different center has the opposite effect, which is a very strong positive feedback. And with this very strong positive feedback, you would then go into have that LH surge or LH peak, or which causes GnRH surge and peak, which will cause then LH and actually even FSH surge and peak. This is going to then be the major stimulus for ovulation. That occurs across most of the domestic species that we're going to study. This is really the trigger. Basically meaning too that, from an applied perspective, from an application point of view, it means that, do you remember when we were describing drugs, or not drugs, but the molecules, and we said that, GnRH, which is a hypothalamic hormone, it's a polypeptide, pretty easy to synthesize, not easy, but very common today. You have commercially very available and actually pretty cheap. So one of the things that, with time, people started to use GnRH was to induce ovulation. So you can have an analog of GnRH inject in an animal, and very, very consistently, if you are in the right stage of the estrous cycle, you can induce ovulation. In certain industries, this was the basis for the for the building of very, to a certain extent, quite complex protocols in which you could basically make this what we call like timed insemination. So you can, instead of waiting for animals to come in heat or to come into estrus and ovulate and then you inseminate, you could do that artificially. You could manipulate the estrous cycle. That will happen for, in order to improve reproduction, you can also manipulate the estrous cycle to actually have the opposite effect, which is contraceptive strategies. So all of that actually estrous cycle that we explained in the previous class, if you look at that, you can, and the reason why you can do it is because you can have access to most of those FSH, LH, GnRH, estradiol, progesterone, prostaglandin. This is all commercially available. So you can modify your estrous cycle in a way or to your advantage, okay? So in the next slides, and we're going to go over again, so at some point we're going to start on puberty, which is, I decided to go first on Estrous cycle because you're going to see that puberty is going to be one of those, I should say, special cases that you could have, you know, within that Estrous cycle explanation. So if you go into basically the slides, you're going to have, yes, the period between two estrous ovulation episodes. We didn't talk about abnormalities, but in the case, when we go to some of the explaining the technologies, you'll see how that can be applied. Yes, of course, it's variable among species. We're going to have a couple slides at the end of this section of Estrous cycle explaining, you know, women have in general 28-day cycle. Most of the large domestic animals will have 21-day cycle. Some small ruminants, for example, they have 16-day cycle. It doesn't really matter. I'm not going to be here asking you how many days the mare has in terms of Estrous cycle, but there is a certain range. If you go into rats or laboratory animals, yes, it's going to be even less. So that's why they can reproduce like crazy. But the thing is, independently of the length of the Estrous cycle, almost always for the species that we're going to talk about, they follow that pattern. Sometimes they have, like in humans, do you remember that you have ovulation, and then you have all those things happening, and then ovulation? So ovulations are basically at the border of the cycles. In humans, it's a little bit different because your ovulation occurs in the middle of the cycle. Okay? Again, the basis for the development of the follicle, positive feedbacks, negative feedbacks are still fully applied. Okay, so you can apply those things from last class to any of those. Yes, we talk about pharmaceutical control. A little bit that we might touch base now is affected by nutrition, production, and other things. Stress, I think we mentioned a few things a few times during class, which is all of those things. And if you think about, you know, all those, you know, you go from GnRH, right, to then LH and FSH, then estradiol and progesterone, and also testosterone in the case of male, but you saw that testosterone we just ignored, you know, in previous class because it's not important. It is important, but it's easy. So the stuff is, all of this will have, and you saw that, you know, they have types of feedback that is mostly negative, you know, it can be negative. This one here can be either negative or positive depending on the concentration. The thing is, imagine that you have this. Now, one thing that we didn't touch base, and we have mentioned in some of the slides, is the notion of seasonality. And seasonality will get in, or the way we can explain is not being specific about seasonality, but we can talk about seasonality, nutrition, stress. The idea is this thing do not work alone. It's not like it is isolated from every other system in your body. So you're going to have things that are external that can affect all of those hormones, but we can focus a little bit on the GnRH. Why? Because some of the other centers that controls other things such as stress, hunger, and feed deficiency and so on, they are really tied to those guys. Light, seasonality, for example, is tied there. So all the way from corticotropin releasing hormone, melatonin, beta-hydroxybutyrate, insulin, glucose, all of this, anything that you can imagine that is either a hormone or a metabolite that affect these other systems, eventually they have some effect in there. So if you are undernourished, one of the very first things that your body shuts down are things that it's non-vital. It's reproduction vital. Well, is super important for the perpetuation of the species, of course, but is it important for you to live? No. So it shuts down really quickly. If you're stressed, your reproduction normally shuts down as well. So this is it follows a little bit of a common sense. Once it goes to the application, you're going to see that, yes, you can probably maneuver that a little bit. You can manipulate light to have, for example, sheep or horses to start cycling earlier during the season. You can do that. You can have different kinds of, probably feeding strategies during breeding seasons in order to improve reproduction. You want reproduction, or you want animals from a body condition score state, obviously not to be too thin, but you also don't want them to be too fat. You have both ends as well. We can go on into this, but I believe most of these questions in more detail we're going to go over in some of the presentations or labs and so on. But it's just to give you this, it's not isolated, everything. And that's where it becomes even more complicated. Okay, so then you're going to see how much more. Then, if you go to a following step, which is, in many cases, we talk about a yes or no. Is the animal becoming more fertile depending on the state of that correlation? But sometimes what you're talking about in industries is like, how can I increase fertility by 5%, by 10%, but just a little bit? Then you're starting to fine-tune. Very small changes. We're not talking about extremes. The extremes are good for us to understand the physiology. But then eventually when you go to the presentations, you're going to see that it's just smaller stuff that we're going to modify. So there is this very nice, slide. You have like lots of other examples in the internet as well that, talk about, the rise and the fall. And I would probably, this is another one, which is not a very good one, but overall, what I would like you guys to do is to go over that little exercise that we did last class. Go make sure that you understand really quickly FSH, then there becomes LH, FSH. As your follicle develops, then estradiol goes up, and then as estradiol goes up, your FSH goes down. It kills all the other follicles, but then you have progesterone, so the bigger follicle also dies, and then it starts a new one. Repeat. After you do that a few times, that becomes really easy to understand. Okay. What we did, and before we go, because I need to go. For next, parenthesis here. So for next week, we're going to have labs. Labs will be in room 342. We're gonna try. We're gonna put this. I forgot this again. I forgot to put the keys of the first quiz on Canvas. I'll put it there, and I'll make a note also for next week's class. It's going to be 342. Yes, if you have a lab coat, please bring it on. You're going to get your hands dirty. Yeah.
Is our quiz going to be in the lab room then?
It can be, yeah. Or we can leave it to the next week. But I think it should be okay to do the quiz in the lab, right? It should be okay, unless you guys feel uncomfortable.
Is it going to be both days?
Excuse me?
Is it going to be both days?
Yes, both days. So that way, we're going to probably try to start, we try to follow more or less what we're given in class. So we're going to start with the anatomy of things, but eventually we're also going to bring, this is gun for artificial insemination. This is what we do when we have to flush the uterus to collect embryos. We're going to bring an ultrasound and say, this is what we see, when we do ultrasound. So we're going to try to give you a little bit of exposure to what is actually done, outside in terms of applications of those things. Is that okay? So I told you this because I need to. So these cycles or these estrous phases, what's going to happen is basically this. So we talk about those, then it goes, and then eventually dies, then another one starts, then select, And let's say this is the nice solid smiley follicle. Okay, once that happens, we saw that, eventually, if we were talking about hormones, the follicles here are developing, FSH is going up and then down. Then eventually here it's probably going up and going down. And it goes something like this, okay? What is going up here as we move is probably estradiol. It's going up, then you kill the follicle, it goes down, and then here it goes up. Nice, very nice. Progesterone at this point, let's say there was another ovulation before here, probably, you start going up, and then it starts to go, and then eventually at some point, it goes down, okay? If this went down, it's because, at some point around here, you have also some prostaglandin. Okay? So this is more FSH, this is more estradiol, this is more progesterone, this is a little bit of prostaglandin. So if you take it like this is a pretty basic, description and illustration of the estrocyte. What this next slides will say is basically like we found, or we humans said, oh, let's do something. Let's divide this thing. So this is what happens to the hormones. This is what happens to them. And this is what happens with the structures of the ovary. One thing that happens is, like, you can imagine that, in most of the cases for domestic animals, each phase will also reflect changes in either behavior or the ability of this animal to, for example, accept mounting behavior, to be bred, and what happens during this next few phases. So what we actually did was to divide this into phases. So we have the estrus, proestrus, metaestrus, and basically that's 3 or 4 of those phases. Once we have this, let's say this early phase here, ovulated, this very beginning of this new follicular, or this new cycle, we tend to call this phase here metaestrus. Okay? Why? Because at this point here, what's going on with most of the hormones? Aside from FSH, let's say progesterone, and lots of these classifications are based on how estradiol and progesterone are present, how present it is. Why? Because they are the ones that have the utmost, let's say, behavioral effect on the animals. So in this case here, how is progesterone? This very beginning here. Very beginning of the phase, progesterone is high or low? It's low. The corpus luteum is basically starting to be formed, so it's not really synthesizing a lot of progesterone. What about estradiol? Like your follicles are just being recruited at that point. Estradiol is what? It's low. Estradiol really is going to start to pick up once you have the dominant follicle going. As the dominant follicle starts to develop, then your estradiol will go up. So this is basically classified, or having this metaestrus phase is known for being low in progesterone, low in estradiol. As you move here to this next phase, which is going to go all the way here, basically. Okay? This phase here is going to be, yes, you have, ups and downs of the estradiol, but there is one that is always up for a longer period of time. Which one it is? progesterone. So this progesterone phase, because progesterone is going to basically dominate that phase and is going to not allow, for example, the animal to ovulate, they have a strong effect in many of the tissues of the reproductive tract. And we call this phase here diestrus. Okay? Okay, so nice. So then we're going to have like basically is one last phase here, but we like to divide it into one, what we call here proestrus. And this very last one here, that depending on the species, it lasts just a few hours or a day or so, is what we call estrus. Proestrus and estrus, they are somewhat similar in the sense that both are characterized by low progesterone, because this happened after you had luteolysis. So your progesterone is then down, your follicles then can continue to grow, so they are releasing quite a bit of estradiol. Okay, so having these large amounts of estradiol will cause a whole different set of changes in the reproductive tract that will be important. For example, if you want to guess, do you think that phase over there is going to be important for what? When you have like very high estradiol, very low progesterone. It would be important for what? First, ovulation, and then after that, probably fertilization. Okay, so you need to prepare stuff in the reproductive tract for fertilization to occur. Especially the estrus phase, this is the one in which up to that point, during the proestrus, yes, you have your estradiol up and so on, but their relationship with FSH and LH is that regular negative feedback. So you're still having low FSH, somewhat, not so much on LH, but you're having this negative feedback on the hypothalamus, in the pituitary. Once they reach that very, very high amount, then you switch. And once you have that switch, this is what the estrus phase is. The estrogenic phase is when you switch the negative feedback to positive feedback in the hypothalamus, and that's when you cause your peak of GnRH and also LH. Okay? Which then will make the follicle happy. Okay? Will make the follicle to explode. It's so happy, it will explode. Okay? So, and then you're going to cause ovulation. That's what it is. Again, for this course, for the sake of the course, this is what is the most important. If you want to go and move over, and for example, we have there, lots of my grad students there, they have to know this, and they have to know by the hour. They have to know exactly how many hours it takes from estradiol to LH, from LH to ovulation, from ovulation to fertilization. Hours. We're talking about hours and synchrony. You remember when I said before about the synchrony of the male and the female? Eventually, for the next class after estrous cycle is fertilization and early embryonic development. And you're going to see that both have to be, lots of nice things happen to the sperm cell, lots of nice things happens after the happy follicle exploded, and the oocyte and the oocyte and the sperm cells, they will have to meet in the oviduct at some point, ready, matured, for fertilization to occur. It's very, very interesting. Nature is amazing in this case, how nicely it was developed in order to create a new being, a new offspring. So in that case, this is basically what it is. So when you go in the slides, so proestrus, you have the development of the ovulatory follicle. Yes, you have high estradiol, low progesterone. If you go into the estrus, they also have high estradiol, low progesterone. The only thing is that still here, development of the ovulatory follicle here is going to be after this stage, it is followed by ovulation. This, another very important thing, visible behavioral signs of estrus. Why this second part tends to be important? One, because we do a lot of research on that. That's why it's important. I'm kidding. But this stuff is like, because this is by how humans started to manipulate estrous cycles of some of the animals. They knew how to breed animals, not by using a male, or let's say when they developed artificial insemination, they had to know at some point when to breed the animal. And that would require a certain knowledge of this, okay? And a certain knowledge of how the sperm cell, how long can the sperm cell survive inside the reproductive tract of the female? How long can the oocyte survive after being expelled by the follicle? And so on and so forth. So you see, you can start adding many layers of complexity into this. So the next phase and the next, so you have, for example, this behavioral signs of estrus tending to be mounted, tends to be in domestic animals, one of the, probably the most classic, signs of estrus. They become restless, they follow other animals, they mess around, they chin rest, on their backs. They become basically very active. They walk around a lot more. I believe in one of the labs, we end up bringing to you a few electronic sensor measurements, because, if you calculate how much an animal normally walks around, and you can basically count that or measure in some way, you would be able to probably measure when this animal is in estrus based on movement only. Why? Because the animal becomes so restless that all of a sudden, and people that are very experienced, either with cows, sheep, they normally identify these animals rather quickly. Why? Because they change. It's a completely different behavior. So that wash of estradiol in the brain really causes not only your generate surge, it changes and it finds other centers in the hypothalamus that changes completely your behavior, your actual behavior, okay? So, which is, it is necessary, for domestic animals. Aside from this, let's give the example of things that we work. This estrus here will last anywhere between 8 and 18 hours. That's it. That's the window in nature that those animals would have to be bred, more or less. When I say bred, is like if they have access to a male, that's the only time the female will allow that male to get even close, okay? After that, kicks and get out of here, and they don't like it. But during those 18 hours, it is. So that's how important the change in behavior is, not only physically speaking, but in terms of accepting others to be close to them. Again, the estrus, first days of the cycle, formation of the corpus luteum, low estrogen, low progesterone, bleeding, and this is we'll go a little bit. Doesn't happen in all species, but this is one of the symptoms that happen when you start having too much of a drop or sudden drops in some of these hormones. Right before or a little bit before ovulation, you're going to see that in terms of timing there, let's leave it like that, but in terms of timing, we're gonna go back to there, because ovulation doesn't occur right after that estradiol goes up. It takes a little more time, but in the fertilization class, we're gonna explain in more detail. But this stuff is Once you have basically drastic changes in progesterone and estradiol, progesterone and estradiol, they tend to be opposite effects on the vascular surface, especially those that are tied with the endometrium. Okay, so sometimes, and the other part is like even the ovulation can cause a little bit of bleeding, you know, especially in very young animals, and you could have a little bit of bleeding coming from some of those tissues. In different species, you can have, for example, during proestrus, the female, the dog, what they have is they can have a little bit of bleeding from the vaginal canal. And this is actually used for monitoring. People would go with the cider brush, get a little bit of sampling from the vaginal canal of the dog, and then go and smear into a slide, look at the microscope and say, oh, there are red blood cells. So not ready yet, not ready yet, until the point where they say, oh, it's ready. Why? Part of this is because once you have high estrogen, you tend to have a little bit of vasodilation. And sometimes you can have a little bit of red blood cells coming into the lumen of some of these organs, either the vaginal canal or the uterus. The other part is like once you have these drops, that can cause a little bit of a vascular effect. So in not very different than menstruation that happens in humans, because this is really like a release of almost the whole endometrium of the uterus because of the sudden drop then, you know, after progesterone has gone down, the end of your cycle, what happens is you lose basically the nourishment of that layer, and then eventually you have the bleeding in that, and all the vascularization that was in the endometrium ends up going along with it. That's why you have the bleeding. There are other kinds of bleeding in a much lesser extent, but again, having to do with this progesterone estradiol changes, okay? Normally, they are associated with that proestrus estrus phase, okay? Every time you have sudden drops in one of the two, you can have some sort of bleeding, okay? Diesters here, of course, basically speaking, you know, go all the way from after this metastasis up until luteolysis, which is functional of mature corpus luteum, relatively low estrogen, you know, not as low as metastasis, but relatively low estrogen and very high progesterone, and associated with follicle turnover. This follicle turnover is another way to explain this up and down without ovulating of the follicular waves. We normally call this follicle turnover. Why? Because it goes without basically never ovulating. So if for some reason or some abnormality, and this is the stuff that, you know, is nice to start thinking about when you have, for example, polycystic ovaries, or you have a luteal cyst, or you have basically cancer of granulosa cells in the follicle, you start going into scenarios where this no longer work. And then eventually I can ask you, so what happens? What's going on? What happens if you have a follicle that, or a CL that never regresses? You would have lots of follicle turnover, which basically means in a production system or in domestic animals, that would be normally very bad. Why? Because you would have infertile animals in the case of milk production, and this is the stuff, without reproducing, you don't have milk production. And in the case of beef animals, if you don't reproduce, you don't generate new offsprings, you know. So this is the whole point that eventually, and with time, we're going to go more and more complex into the application of it. Okay? So we mentioned a little bit about this seasonality we mentioned a little bit, not so much on the postpartum. One thing that there's another, and this, we can tie this a little bit with the puberty side, which is a little bit of a special case. And going a little bit, some of these things, and most of these things that we're going to talk about is non-pathological. It happens. When I say an estrous would, some people don't even like to call an estrous, because an estrous would imply no cycle at all, nothing going on, which is not the case. It's very rare that you would have like zero activity in the ovaries. You're always going to have, even if it is a little bit, you can have a little bit of recruitment, die. A little bit of recruitment, die. But zero, very, very, very uncommon. So sometimes they like to call this a anovulatory condition. And this anovulatory condition, think about, we can think about animals that are non-seasonal, meaning they can cycle through the whole year. Humans are like that, cows, and let's put these two examples. Basically speaking, they don't have, if thinking in a after puberty and until they get pregnant, they just cycle, ovulate, go back, cycle, ovulate, go back, cycle, ovulate, go back, and so on. In the case of seasonal animals, for whatever reason, it can be the most common for us in terms of seasonality is going to be like dogs. They basically have one cycle every so many months. So they are able to be pregnant maybe twice a year or so, a little bit more, a little bit less. And sheep and equine. Sheep and equine, they tend to be opposite. One, as light goes up, they tend to start to cycle. And the other one, sheep, as lights go down, then they start to cycle. That's because melatonin have a pretty big effect. We can talk about this a little bit later. But basically speaking, the idea here for this slide is as they go into this anestrous phase, what happens? It basically goes to a point where almost like pre-puberty. They just have the follicular development, but it dies. Follicular development, it dies. Follicular development dies. And then why? Because something there is going or having a negative effect on all that GnRH, LH, FSH, progesterone, estradiol axis, okay? Basically, no expression of estrous behavior, no ovulation, no corpus luteum formation, and high levels of follicle turnover, okay? So in that case, you can have high levels of follicle turnover in many different ways. In that case, for an estrus or for seasonal breeders, for example, it has to do with basically not having the right stimulus, in the hypothalamus to continue the cycle. This case, for example, of postpartum and estrus, we're going to have a class only on that at the very end of the course. But basically, just as a preparation, after pregnancy, you can imagine that you're going to have a few, up until you have your uterus basically going back to a normal size, you're going to have a time in which your ovaries will not function normally. So you don't start cycling a week after there was a parturition. So in most of the domestic animals, that doesn't happen. But it has also a lot to do with the stimulus from the mammary gland. So nature found a certain way to say, listen, if you're still doing certain things with your offspring in terms of, lactating or other things, they tend to block signals or send a signal to the hypothalamus saying, it's not yet time for you to start cycling again. Let's focus your efforts and energy to the offspring and not necessarily to give birth or starting the reproductive cycle for another the next offspring. So obviously, you can imagine humans try to manipulate that a little bit in order to have animals to cycle a little earlier and maybe to have pregnancy a little bit faster. Okay? And obviously, what you also have is trying to select animals that can have that. So you see lots of these little details, little things. Eventually, there's lots of use. I think in this case, there is all the way from genetics to tools to, for example, identify estrus, to manipulate the estrus cycle. There is a big industry around those things in most of the domestic animals. So this will be eventually applied to many, many things. Okay, so this is just to say, so normally you all the way, if you think of a life of an animal, the animal goes into a certain estrus for a long time, and then puberty arises or arrives, and then you start to having these cycles until you get pregnant. As you get pregnant, implantation, gestation, parturition, lactation, and then you can go into or go back to this, for example, an estrus, or go back here to the estrus. So this is the case, for example, to many species such as ourselves, cows. This is monoestrus cycle is one example that happens, for example, with dogs. So they don't, even a few months after parturition, they don't necessarily get back into estrus cycle. They just spend a long time in an estrus, not caring about reproduction, and then eventually they come back again. Again, this name, monotocous or polytocous animals, this tends to give only one offspring per time. So they tend to ovulate one. Ewes, I put an asterisk there because sheep, you have some breeds that are really have been bred through the years to genetically be more like even twins. So sheep is very common to have twins. In the other species, mares, it's extremely rare to have twins. Humans, somewhat. Cows, you can have it, and in some of the cases that you're going to see in technology, this thing here, this process here, can be affected and somewhat induced to have more twins or more, or what we call double ovulation. So 2 follicles ovulating instead of 1. And again, those become fine-tuning, fine differences between what one, and we can actually not promote, but we can induce somehow that to happen. Induced ovulators, so mostly are spontaneous ovulators, which basically means we have to wait until this estradiol goes really up, and then to promote or to induce a GnRH and LH. That's what I mean by spontaneous. So you have to wait until that happens. Induced ovulators, cat and mice, and rabbits to a certain extent too, sometimes what they do is like they ovulate once they have some sort of stimulus. And normally this has to do with neuroendocrine pathways. And by neuroendocrine pathways, meaning sometimes you need to have some stimulus, for example, in the vaginal canal, or stimulus in the cervix, which basically in general means during copulation for that to be induced. Okay, so that's another way to do it, but that's a much simpler way. So instead of, they still have that, but they don't necessarily only depend on that. Normally they have to have the stimulus of the breathing process in order for ovulation to occur. Okay. Oh, how that happened, Ronaldo? What do you mean by, stimulus from the vaginal canal, the cervix? The same way that you have, if you have stimulus of the mammary gland, or for example, a calf suckling into the cow, that stimulus is what actually drives the milk let down. So the stimulus in the mammary gland goes through a neural pathway, so there are nerves in there, goes to the spine, goes to the hypothalamus, liberates, for example, oxytocin, that lets the milk down. This is the same. The stimulus in the vaginal canal or cervix go to the hypothalamus, go to generate centers, release LH, FSH, especially LH, and that causes ovulation. Does that make sense? Yeah? Very good. And then we go, again, important here to have this distinction. The only way I would ask you guys is not to know or not to memorize this, which of the species have. Well, what I would like you guys to do is at some point I ask you, is it possible to have more than one ovulation? Is it possible to have multiple ovulations? Is it possible to stop, ovulating for a certain period of time? And then what I would like you to do is to think on things that it can happen. If I stop this, it means that, light can come in, or nutritionally is bad. And I want you guys to think about it. It doesn't have to be necessarily the right answer, but what I would like you guys to do is to play with that. And as you play with that, you're going to see that more and more information will be needed for you. Okay, so if I tell you that, these animals here, they are induced by light and they stop, cycling. What does that mean? How do you think it happens? So I'm going to give you enough information for you to play with. So not necessarily knowing all of these things. Same one for the next slide. So you have spontaneous, induced, you have seasonal, no seasonal, so on and so forth. So not a big deal. It's just explaining, how diverse. And if you go to wild animals, then this goes bananas. Okay, then this goes crazy because they have even more extreme, strategies for reproduction. If you go to invertebrates, then it goes even crazier. So that's how the thing goes. Okay. Why do we have luteolysis out of nowhere? Let me see. I think the point, eventually, the reason, and I think I mentioned before, is that all of these things, they seem all hypothalamic, pituitary, gonadal, and the only hormone in this whole thing that is outside this axis really is prostaglandin. And the idea for luteolysis, now I remember. The stuff is that luteolysis tend to be something of a special importance because this point here, and then later we're going to go into more detail, but this point here is really a very, very important point in the cycle because this is where later in the course we're going to have something that will decide if the animal becomes pregnant or not. Okay? Normally, if you're not pregnant, or if there is a very underdeveloped embryo, this is what happens. Meaning, your prostaglandin will be released. And with the prostaglandin being released, progesterone goes down, follicle continues, ovulates, new cycle. So you continue the cycling, the cycling cycles. Okay? In the case of pregnancy, the baby will try to block this. Why? Because later on you're going to see that, of course, progesterone will be extremely important for the maintenance of gestation. So normally this is the case where you can go to cycles or we can go to gestation, which is going to be the second half of the course, all about the gestation side. And understanding how this luteolysis work is extremely important. So this determines the length of the diestrous phase and the estrous cycle, which is very true. In most of the species, the reason why they have 16, 21, or 28-day cycles has to do with when luteolysis occur. Okay, it basically mandates. Can you manipulate this thing? You can. okay? After knowing how this luteolysis works, yeah, you're going to understand. It's the major luteolytic agent in most domestic species. It's funny because most of the species will have different strategies to block this. Like every embryo from different species synthesize some molecule that blocks the prostaglandin synthesis or diverts prostaglandin synthesis in different ways. But prostaglandin F2 alpha tends to be the major luteolytic molecule for all these domestic species that we normally talk about. It is synthesized in the endometrium, in the uterus, basically and more specifically in the superficial epithelial cells of the endometrium, not in the glands of the endometrium, but not in the interstitium of the endometrium, but is in the very, very unicellular layer of cells in the epithelium of the endometrium. And then you can say, how does that make any sense? What is the point here of, well, later on you will see that contact of the baby with that layer will be one of the most important things to block the effect. Okay, so you're going to have part of this blockage of prostaglandin synthesis by the baby that we, generally speaking, we call this phase maternal recognition of pregnancy. Is done by paracrine defect, not endocrine, not autocrine, paracrine, cell to cell. Okay, so again, for a different class, but just to keep that in mind, you know, there's always a reason for things to happen the way it does. Synthesized by the uterus, obviously countercurrent transfer to the ovary, plexiform structure, very well, you know, described last class. And the stuff is like the next one, and obviously inhibited by the conceptors in pregnant animal. So if you go here to, I'll be back to the other slide. So this is an even better slide than the other one that I had. But basically, it has reinforcing. This has to happen because if the prostaglandin go to the general circulation first, it's going to be metabolized. It is normally metabolized by the lungs, okay? And is very quickly. One turn into your lungs and 90 to 95% of this prostaglandin will be metabolized into, obviously, a metabolite that doesn't have the same effect, okay? So it has to go first. Oh my God, it's going to the general circulation. No, it goes back to the ovary. It goes back then to the ovary, and then it goes through the general circulation. So prostaglandin can then bind to receptors that are in the corpus luteum and do what it does, which is normally to induce apoptosis, block steroidogenesis. So you're going to have stop like the blockage, both the structural and functional of the corpus luteum. Does that make sense? Yeah? Yes? Good? Nice. So this is a cross section of a corpus luteum. This is the rest of the, you know, some of the ovary structures. This is the medulla. Sometimes you can have a little vessels or little follicles, but basically speaking, you have this structure being in the middle here. It's very yellowish. You know, when you see in lab, you're going to see a few of this corpus luteum. It's like a big ball. It protrudes from the ovary, such as the size that it forms. And as I said before, it goes into basically all those thick cells, granulosa cells. They rearrange into what we call large and small luteal cells. They go from estradiol-producing cells to progesterone-producing cells, and they also exhibit this prostaglandin receptor. Prostaglandin receptor is there. They express this receptor, and after they are and if it is induced in the right way by the uterus at this moment, and normally this is only, again, this is another, we're always talking about the synchrony for fertilization, for this estradiol, LH, ovulation, and on the other side, the sperm transport and all this stuff that has to be super synchronous here in order for offspring or fertilization to occur. This part here also requires a certain synchronization. Why? Because the synthesis of prostaglandin really bumps up at a certain stage here. This stage here, it's really when you are somewhat here at the end of the estrus, but it also has to synchronize with a certain development phase of the embryo, because only at that stage is like the embryo can produce something to block the synthesis of prostaglandin. So I think with that, I think one of the nicest things about reproduction is that, yes, it is somewhat inefficient in the bigger scheme of things, but it is inefficient because all of those things, we haven't talked much. I think today was the very first class where I talked so much about timing, synchrony of things to happen, because even if things go well, if they don't overlap at the right moment, either the luteolysis or blockage of luteolysis and fertilization and ovulation, and then you're going to see passivation from the sperm cell. If they don't occur exactly at the right time, it doesn't matter. You won't have what is the major thing that you would like to have, which is the generation of a new offspring. Does that make sense? Yeah? So small, large luteal cells, they do have these prostaglandin receptors, which is a long-chain fatty acids, a membrane receptor. I think in the last, forget about this portion here, just for a second, nice. And then, but basically what happens is you promote apoptosis, you block angiogenesis, so you block circulation, you promote apoptosis, and you shut down the steroidogenic enzymes. So you basically kill the gland in any way possible. That's why you can go from that big protruded gland that you're going to see in lab to basically a little scar. Okay? From producing lots of progesterone to basically producing zero. Okay? Okay, so this is just a little slide for you to have this sense. Basically speaking, for most of the species we talk about, domestic species, 3 weeks, again, take a look at the range, but 3 weeks around that tends to be the length of the estrous cycle. You can have several of those estrus or these follicle waves within the estrous cycle. It depends a lot on the sensitivity of each animal to some of the hormones, so you can have more or less. Why? And this is the good hook for the next portion of the class, which is puberty. Basically speaking, of course, so this, I said, you have all this estrous cycle. Aside from the mare, most of the other species, they have not very long estrus length. So again, talking about synchrony, that's the time they have for breeding or for copulation. And time to ovulation, this we're going to go afterwards, but basically speaking, after you have estrus, your ovulation will only occur several hours after that. And that will be required because if that doesn't happen, for the most part, you're only going to have, by the time you ovulate, your sperm cells have to be there already, ready. That's why it requires a little bit. If estrus is a time where the female accepts the male, and ovulation just occurs somewhere in here, it means that the sperm cell will be deposited here, and it will stay around for a few hours. It is required. We're going to see in one of the other classes that, you know, the sperm cell needs to be inside the female tract for a few hours for maturation. And then ovulation occurs. The oocyte is a little bit different. is ready to go. Once it ovulates, is ready to be, basically ready to be fertilized, but they have a very short lifespan. So most of the time, the limitation there will be the oocyte. So by the time the oocyte enters the oviduct, the sperm cell has to be there, ready, mature, little Michael Phelps, go, and ready to fertilize the egg. Okay, that's that point when they are somewhere like very close to each other. That's where swimming, the capability of the flagellum to actually propel, to make the forward propulsion of the sperm cell, that's when it's going to matter. All that way from vaginal canal to the oviduct, it is not basically done by the sperm cell. It's done by movement of the female tract. Okay? Very, very good. So now, instead of going there, we can go and go to the beginning of the class, which is puberty. And the reason why I said puberty, like after knowing all of this, have a sense of that to happen, luteolysis when it happens, Then we go on to say, or to talk about puberty. Look at some of the classes on folliculogenesis. I said, yes, you are born with a set number of follicles, which means a certain number of oocytes, a certain number of eggs. Eventually, you're going to have those follicles being recruited, and eventually ovulate, and sometimes do not ovulate. But before puberty, what happens is this continuous cycle of growth and atresia, growth and atresia, growth and atresia. It never reaches the final stage. Okay. Now, from a definition point of view, puberty, when you reach puberty, it means that's the time when you had the first ovulation followed by a complete estrous cycle. In the case of males, it was the time when you first produced mature, fertilizable sperm cells. That's the definition of puberty. And basically, it has to do with releasing the egg or producing a fertilizable sperm cell. That's basically what it is. Now, age at first estrus followed by ovulation has a big, one of the biggest, is a little bit more complex. It's not more complex. It's the system or the mechanistic side is basically the same for males and females. Puberty also has, and I put it like a special thing on puberty because puberty tends to be something of very high value in commercial systems. Why? If you reach puberty earlier, it means that you can reproduce faster into the system. So if you have an animal, and in my case, I'm much more familiar, you're going to see with cows, there are breeds that tends to reach puberty, for example, especially tropical breeds, tends to reach puberty with 16, 17, 18, 24 months. So they take 1.5, 2 years until they reach puberty. Sometimes it's not even dependent on a plane of nutrition and other things. They just don't reach maturity, sexual maturity very early. Some other breeds, especially some of the smaller European breeds, such as Angus or Hereford, especially the British breeds, they tend to be very precocious. So by 9, 10 months, they already, even before 8 months, they already reach puberty. So from a systems point of view, you're almost always, aside from the fact that you could have puberty too early, and what would be, for example, from a commercial standpoint, what would be a problem of reaching puberty too early? Not a problem, but not so much of an advantage. Yeah, so you would have an underdeveloped, not so much the reproductive system, but it would be actually their, if I put in simple words, size. In order for you to reproduce, you have to have a certain size. And that has to do with the birth canal size, would have to do with actual bone structure for that to happen. So it doesn't matter. You can have a cow that, reach puberty super duper early, but that's not so bad. That's normally not the case. Normally the case is like, yeah, sometimes they reach puberty at an age that is not necessarily the best for breeding, but you made sure that they are sexually matured by the time when you're ready to go. So the earlier, commercially speaking, the better, okay? Now, it's variable among species and breeds, of course. Now, this is the part, the other slide we'll try to go a little bit over it. We started, and remember the first phase of previous class, I started drawing this part, and eventually, I think it was this follicle that ovulated. I was like, no progesterone, there's just this little thing. You can imagine you're going, from that guy until that guy ovulated, you were doing like this. Yeah, and then again, and it's not going. Oh my God, it's not going. And then it's not going. And then it's that. And then eventually, let's say you ovulated here. Knowing what you know about this, what do you think it might have happened? What do you think it could have changed in that cycle to eventually the cycle be able to ovulate? Because that's what it is. You're gonna have to go into puberty doesn't occur in a all or nothing kind of thing. It occurs with a process. Because even before puberty, you do have those cycles, those follicular turnovers. Okay? So if you have to think, let's say this, strong negative feedback on estradiol. What do you think that means? Estradiol will have negative feedback on what? SH, some GnRH, some LH, but strong on FSH. Let's say, on that. So what do you think happens? I think when I say strong negative feedback, the reason is it as you mature, as you age, let's say you produce, remember here that, this guy here starts to produce quite a bit of estradiol, and that's when you have this actually FSH, negative feedback. What if here it's already enough to kill those follicles? What it is if it is already here, basically meaning here you need plus of estradiol. Here you need plus of estradiol to produce the same negative feedback. Let's say here you produce just a little bit of estradiol to promote the same negative feedback. This is what they mean about strong negative feedback. It's almost like your hypothalamus and your pituitary are very sensitive to estradiol. Does that make sense? So anything that is produced by the gonads goes back to the hypothalamus and shut it down. So as the follicle starts to develop, it's shut it down and shut it down and shut it down. So you never reach a level that is high enough to cause ovulation. You never reach a level of estradiol that is high enough to go to the different center of the hypothalamus to then cause the GnRH and LH surge. Okay? As it moves, then you have basically both gonad and hypothalamus. One is your gonad's or your basically your Leydig cell producing testosterone, or your ovaries, your follicles producing, your ovary in general producing estradiol, they start to develop, they grow, and they start to slowly producing a little bit more of estradiol and testosterone. In the case of testosterone, it's a little bit different because you do not need the peak eventually, but you need a certain amount, in order for spermatogenesis to occur. Eventually, this decrease in negative feedback occurs. A lot of that, this decrease in negative feedback, you can translate into what? Let's say you have just a little bit of estradiol that can produce a lot of negative feedback in the hypothalamus. What do you think is different in the hypothalamus in this prepubertal stage? What do you think it can be different in that hypothalamus? When you say hypersensitive, describe to me what hypersensitivity might mean. More receptors, 5 point. So that's the whole thing. You have way too much. So any ligand that is circulating attaches negative feedback. As that amount of receptors starts to vanish, and you have a little less, and a little less, and a little less, that same amount of estradiol will cause less negative feedback. If it causes less negative feedback, it means that that follicle can grow a little bigger. It is producing more. So eventually, you start to produce more estradiol, and your hypothalamus start to have less receptors for it. So that negative feedback keeps decreasing up until a point where the estradiol can reach a concentration that is high enough in other centers and then cause the GnRH and LH surge. This is from a physiological point of view, from a hypothalamic gonadal point of view. This is what puberty is. It's basically reaching a point where your hypothalamus and your gonads can almost control themselves or have a less sensitivity to each other to at some point cause ovulation. Okay? Yeah, this is basically one of those pictures. Not the same. It's like very, it shouldn't be that way, but basically speaking, before puberty, you would have no LH. After puberty, you start to have more LH, but it describes, like this one here, I think it describes a little bit better. So you have before puberty, you have your pulses of LH or GnRH are like this. Now you start to have pulses of LH, of GnRH that are much, much higher as you move on. So your estradiol your negative effects of estradiol are much lower, and you have more peak of LH, more and more, until you reach the actual peak of LH. Okay, this is not, okay. Hormonally, this is, basically speaking, what happens during or affecting the initiation of puberty. Sometimes you can have reduced GnRH release, pituitary response, ovarian response, but this is normally a question of variability among animals, okay? If you have animals that are a little bit more, when I told you before about the changes or the differences in breed or species, is because they can have different sensitivities to it. Can other things affect that? Yeah, everything that I said before that could affect the regular estrous cycle can actually affect reaching puberty. So nutrition, if during the growth of the animal these animals were, for example, undernourished, yes, you're going to have a later puberty. You're going to achieve puberty a little bit later. So breed, genetics, crossbreeding in general, in most of the species will tend to make your puberty goes a little faster. So, but that's, we won't touch much of these in this. Environmental temperature, I said about nutrition. Temperature and humidity has a lot to do in the same way as confinement or presence of a male. Lots of those things, they have to do with stress. If an animal is under, for example, heat stress or any sort of stress, generally speaking, puberty will be delayed. Okay, so anything that can affect your pulsatility of GnRH and LH will eventually be negative to you, or negative for reaching puberty. What is this? Let me see. Let me go just to this. Let me see here. Mentioned before, yes, you can induce puberty either through genetics, either through manipulation of certain things for economic advantage. Especially these seasonal breeders here, sometimes you do that in mares as an example. Sometimes, you know, you can expose mares to longer daylight or light cycles. So even if, you know, environmentally you are in that, let's say, 10 hours a day, 14 hours a night or something like that, what you can do is you complement. So you put them in like a barns that has a lot of light, and that can hasten or can reinduce not only starting to cycle again, but also puberty. You could give a little, what you can do in those cases is to give a little push for that hypersensitivity to go down quicker. At the end of the day, that's the basis for it. This is just a comparative event of male and female. Give it a quick read, it's pretty nice. And then this is age of puberty in lots of the species. So you're going to see that varies a lot, you know, on the species. But if you talk about bovine, canine, equine, feline, ovine, porcine, lots of these, especially those domestic animals that we're used to, you see that anywhere between 8, 9 months to a year, it's where lots of these animals, they reach puberty. In the next slide, you have actually by breed. And then you're going to see that, yes, it varies tremendously depending on the breed that you have. And that has a lot to do with types of breeds, where they were selected first. You know, if it was more for temperate climate, if it was for tropical climate, and that goes for dogs, sheep, swine. And this is where a lot of the economics go. Mix breeding those animals, or trying to see a lineage within each breed that can be reach puberty earlier, or if you extend that selection, can be more fertile at some point. So this is just a little differences in kind of a detail, less to do with, I wouldn't cover this, but just so you know, people manipulate that a lot. Okay, very good. So next week, classes, labs, and let's see. Are we gonna cover all of it? So what do we have? So first one was only anatomy, right? So then what was the other classes that we had?
Hormones, what type of analysis?
Yeah. So we had hormones, and then, oh yeah, no. We don't have to go to Estrous. So hormonal processes and gametogenesis. Okay. No puberty, no estrous cycle, at least for this one. Is that okay? Yeah. We're putting. Yeah, I forgot for the last time. Okay.
If it's not up, can you send me an ad? Yep. Okay. Yeah, no, guys. Oh, perfect. If